I've been around this block a few times. I know how this works. Okay, so yeah, right. Could you repeat everything you just said? Yeah, no. Okay, so let's go ahead and pick up where we left off. And I'm aware that I haven't posted the video from last time yet. After this one gets processed, I will post both of them together because I know that our exam is next Wednesday, our lecture exam on this. And so that means we're deciding the cutoff is going to be the end of today. So wherever we are at the end of today is going to be our cutoff. And so make sure you sort of draw a mental line in your notes at that point so you know what's going to be where. So we're talking about our coronary system, right? We just got done talking about the arteries. We're talking about the right coronary and the left coronary and the branches of those two guys. And so that's basically feeding blood to the heart. Now, what about the venous side, right? What about when your heart is done feeding or doing this thing and it's got to basically drain that blood through the veins? Well, the venous system is basically going to be, or the arterial system is mirrored by the cardiac veins on the venous side of things. So a couple of the biggies that we're going to see is clearly, first of all, the great cardiac vein. This is the one that's going to be mirroring the AI, the anterior interventricular. And so you did basically see them like right together, like really right next to each other. And so the important thing always to understand is that when you're talking about the cardiac veins, regardless of where you are, whether you're on the anterior side or the posterior side or whatever, they're all basically draining into and moving toward the coronary sinus, right, which is kind of like that little swollen area on the posterior side of the heart that kind of represents sort of a collecting area before the blood all moves into the right atrium. Now, there is also a series of anterior cardiac veins, smaller ones, but these guys are going to be emptying directly into the right atrium. So it's kind of a little bit different in terms of that particular sense, in terms of how you're routed and directed. But a couple of the other biggies that we're going to be taking a look at, and these are the reason why I call these biggies is because I'm looking for pairs, right? So we already talked about some pairs, some arteries, AI, the posterior interventricular, the marginals, right? So we want to take a look at the veins that are mirroring those guys. We've already covered great cardiac vein as mirroring the anterior interventricular. The posterior cardiac vein and the middle cardiac vein and the small cardiac vein are all going to be mirrors for like the marginal, the posterior interventricular, and so forth. And so these guys are going to be important because they're going to basically be draining the blood from the exact arterial branch we talked about last time. And then, of course, these guys are all moving toward the coronary sinus. So pretty much mostly, for the most part, the overall theme is have a vein that mirrors the artery and basically drain it into the coronary sinus. That's basically what we're looking at. And so here's kind of an image. We're going to have to rotate this a little bit. Sometimes it's a little difficult unless you get the right angle on it. You can see here clearly, you can see the origin of your coronaries, right? So here's your left coronary coming right off of that conus arteriosus area, right? So that basic, that kind of, that swelling area at the base of the aortic, the ascending aorta. And then that's going to branch immediately into the AI, which is this guy right here that goes down. And that's going to be mirroring the interventricular septum, and of course the circumflex, which kind of wraps around the underside of the left atrium. Now, mirroring the AI is going to be your great cardiac vein. So those are the two pairs on the anterior side, tracking along the interventricular septum. On the right side, you can see the right coronary, which is originating at about the same place on the other side of the ascending aorta. And so the right coronary is going to come out, and it's going to branch off into all sorts of little branches. And each of these little guys has names to them. You're not going to learn them. If there's a cardiology class, you would, but we're not here. We're just going to look at the main ones. So these ones that are following these smaller branches, those are those anterior cardiac veins that we mentioned before on the previous slide. Then as you kind of come down, you have the marginal artery here, which is mirrored by the small cardiac vein, right? So the mirror of this marginal is the small cardiac vein. And then as you turn this around, now what we're looking at is the back side of it, the posterior side of it. So here is going to be the other margin. So one of the marginal arteries, which is the other side. So here's your small cardiac vein coming around. So what we've done is we've rotated it around So we looked at the right margin. That's what this guy is right here. This is the right marginal artery over here. And this one up here is the left marginal artery. So this left marginal artery is being mirrored by the posterior cardiac vein, and that's draining blood to this coronary sinus area. And then the posterior interventricular, which is this big old guy right here in the middle on the posterior side, which is also following the septum on the back side of the heart, the posterior side of the heart. This one's being mirrored by the middle cardiac vein. So when you study these as pairs, it becomes a little bit more, it's a little easier to study them together as pairs because that's kind of the way they're wired. Awesome heart. I wish we could actually figure out how to get a hold of this thing, but this is probably one of those preps that some wizard guru in a university somewhere did special for this particular author. And so this isn't something that is in general distribution that you can just like order from like an outfitter. Trust me, I've looked for it. Doesn't exist, but it's really cool because one of the things they've done is they've really cleaned this up, right? So they've basically taken and they've left a musculature, but what they've done is they've sort of treated it, sort of dissolved, if you will, all the fat and stuff like that. And then they injected each of them with latex, just the heart region. That's the reason why you see the blue and the red. Now, would it be nice to actually have an actual version of this in the lab? Yeah, it'd be awesome. Unfortunately, to get a heart that looks like this, that's this clean and this well injected, is probably going to be astronomically expensive and probably pretty limiting in that sense. So here you can see, actually, this is an actual human heart. So you can see it's that we're not lying to you. This is actually a real, this is actually real, right? So here's your coronary sinus right here. So you can see it's kind of nice and pooped out, and you can actually see how your veins and your arteries are basically as billed, right? So here's your posterior interventricular. If you were to turn this around, you'd be able to identify all those pairings. Okay. So the way we're looking at it here is basically edge on. We're looking at the left edge of the heart. That's the reason why right in the middle you have your marginal artery. This is your left margin over here, looking primarily at the cardiac vein situation. Okay, let's take a look at the conducting system, right? That's basically what we're going to be going here. So that's a lot of anatomy. And now what I want to do is I want to take a look at some physiology, right? So now we're going to be looking at some of the mechanics and some of the way that the actual heart is working. And so in order to understand heartbeat, we need to understand, first of all, a couple of things about how the heart contracts, right? So basically the heart contracts essentially in series. First, the atria contract, and then the ventricles. We covered that last time, right? So that part we kind of know. But in order to actually get this to occur in sequence like that, as opposed to everybody doing it all at once, we have to basically make sure we wire this in such a way that that communication gives us our desired result. The idea being then we have a very desired way that we want the heart to beat. So when we set up the electrical, we have to make sure that it's wired to give us exactly the outcome or the effect that we want. And that's kind of what the heartbeat and the conducting system is all about. It's now that you have the structure of the heart, how do you wire it so that you can get it to do what you want? Okay, that's what you're doing. So when you look at the physiological aspects of cardiology, the one thing that you need to understand, it's very important, is that there are actually two different types of cardiac muscle cells. We didn't tell you that in AMP1, did we? Surprise. Not that we lied to you, but cardiac cells. The first one, the one that we told you about, were the contractile cells. I'm going to start off with the second one first, right? So this is basically what we know. So this is basically a standard, normal muscle cell, right? Just like you learned about in AMP1, right? So we learned a little bit about that sliding filament thing. Remember, cardiac muscle cells are also striated, which means they've got sarcomeres, right? That basically means they've got that sliding filament idea. And so they're contractile. And this is basically what's causing the heart to collapse down, or essentially to squeeze the pump so that you can actually shoot blood out of the heart. However, there's also another type of cardiac muscle cell that's actually quite different than what we usually think of as muscle cells. And these are conducting cells. So these guys are actually conductive muscle cells. It's like one of those hybrid cells. Remember, we ran into that before, didn't we? In the endocrine system, we ran into neurosecretory cells. These are neurons that secrete. So you have kind of a combination or fusion of neural tissue and epithelial tissue. Now we have another fusion, kind of another hybrid, if you will. It's a fusion between the contractile tissue, which is muscle, right, and the conductive tissue, which is neurons. So in this case, instead of having a bunch of neurons in there, what you actually have are real, honest to goodness, muscle cells who have the capacity to conduct a signal in the same fashion that a neuron can do. Now, these guys are in control, and there's not as many of them, so these are less abundant by a lot, right? But these guys are designed to control and to coordinate and regulate your heartbeat. And so those are important because who's listening to the signal? Your cardiomyocytes or your myocytes, right? So that's basically the contractile cells. But who's sending the signal? Your other cardiac muscle cells, the conductive ones, right? So this is kind of like a command and control flow. It's like a chain of command, if you will. Okay. Now, when we take a look at the cardiac cycle, which we'll be taking a look at here shortly, it's really beautiful in all of its output. But basically, we want to start off with in the conduction system is with a patch of cardiac muscle cells. These are those conductive ones that form what's called the SA node. The SA node is essentially refers to the sinoatrial node. So that means it's in the atrium. It's actually in the right atrium. And so what happens is these are the conductive muscle cells. So any kind of an action potential is going to go through these conductive muscle cells. That's going to be transmitted through a more larger and extended conduction system. By the way, this shouldn't be foreign to you because in the nervous system segment of AMP1, you learned about action potential propagation and disseminating it through a network of conducting cells, in this case, neurons, right? Same basic idea, if you think about it, when you look at neuromuscular junction, this shouldn't be too foreign to you because we already know that muscles are conductive, right? How did the action potential actually reach inside the muscle cell? It had to depolarize the muscle cell's membrane, right? The sarcolemma. That means that the muscle cells inherently are themselves subject to action potential. That means they can propagate action potential. All right, this is just an extension of that. Okay. Now, what this does then is as this is moving through the conductive muscle cells in association with the SA node, these action potentials will then reach the cardiac muscle cells, the contractile ones. And then that will, just like with the neuromuscular junction, that'll tell those. Now, how do we measure this? Think about it, right? If you're sending electrical impulses, which you are, then how do you measure what's happening? You can, right? Because you can measure electrical impulses. We did it in AMP1. We just didn't do a lot of it, right? But that's a physiological thing that you can do. Whenever we were measuring changes in membrane potential during the contraction cycle, that was us measuring the voltage changes of what's happening inside your neurons. So we can essentially do the same thing with an electrocardiogram or an ECG or an EKG. This is basically a voltgram. That's not an actual thing. That sounds a little thuggish, but basically it's a voltage read of the potential changes during the cardiac cycle. Because these are electrical events that are occurring. These are action potentials that are moving And so that's what's underpinning all of the stages of the cardiac cycle. So we can record those then with an electrocardiogram as a change in voltage or a change in membrane potential. That's essentially what an electrocardiogram does, or what a voltmeter does. It changes differences in voltage. So when you take a look then at the conducting system overall, the real kind of secret sauce to all of this is those specialized conductive cardiac muscle cells. This is unlike anything else that you have in your body. It's also unlike any other muscle cell that you have in your body. It can initiate and propagate electrical impulses. The only place you've been able to do that was in your nervous system. This is clearly not your nervous system. So it's borrowing heavily from what the nervous system does for a living. Now, the other important thing is automaticity. When an action potential is rumbling down an axon, it came from somewhere, didn't it? Did it just happen spontaneously? No, usually there's some sort of a signal that created it, right? That kind of tripped that up, and then you started your action potential. So generally speaking, whenever you get an action potential in your nervous system, it's being communicated by somebody else, somewhere in your nervous system, in your central nervous system usually, and that's basically stimulating these action potentials. So they were basically created by something. But the thing is, the SA node contracts automatically. That's what automaticity is. So basically, this is the self-initiation of the SA node cells to create, I'm going to use the Latin term, de novo, an action potential. What does de novo mean? In Latin, it means, you probably heard that term, right? But you didn't know what it actually means. In Latin, it actually means from nothing. So to do something de novo means it doesn't exist and poof, it exists now, right? So it's like magic. Your nervous system doesn't play that way, right? We even learned very painstakingly that even when you get an action potential, you see it coming, don't you? Because you kind of get that little rise in action potential, that graded action potential that's sub-threshold, and you kind of see that slowly increasing over time until it reaches threshold, and then all hell breaks loose, basically, right? But we see it coming. In this particular case, you don't see it coming. It's automatic, and it comes from nowhere. Nobody's telling the heart to beat. The heart is just spontaneously doing it on its own. That's what automaticity means, which is unlike any other system in your body. Every other system in your body is being told what to do by your nervous system or by your endocrine system. The heart is the only one in your body that's doing its own thing without being told by anyone, right? So it's very much like a metronome. It just beat, beat, beat. It's like, well, who told you to beat? Nobody. I just beat, beat. I'm just like on this metronomic pace. That's what I do. Once I start it, I don't stop it until you're dead. So this thing is happening for like 80 some odd years, or however long you live. It's automatically just, it's like a perpetual motion machine, right? It just keeps going. Nobody's telling it to do that. Your nervous system isn't telling it to do that. Your nervous system actually tells your lungs to breathe. Most of your autonomic things are being told to do what they need to do. It's just that they're happening in the background so you don't think about it. But the heart is not being told what to do. It's not being told to strike a beat. It's doing that on its own. And nobody's talking to it. There's nothing like that anywhere else in your body. And so that's that automaticity piece of cardiac musculature. So let's take a look at some of the major parts of this conducting system that are kind of introduced softly to the sinoatrial node, the AB node, right? So this is going to be a patch of cells, those conductive cells, right? Those conductive cardiac cells. It's going to be in the wall of your right atrium. It's going to hang out. And then another node that you have part of your conduction system is what's called the atrioventricular node, or the AV node. So this is actually between the junction between your atria and your ventricles. So this is kind of at the bottom of the atria as they touch or meet the ventricles themselves. And then also as part of your conducting system, you have a whole bunch of different conducting cells scattered throughout the myocardium. They basically permeate myocardium in order to distribute the message. The message is contract, right? So that's basically what you're doing. It makes a lot of sense, right? If you're going to send a message, then you want to make sure that everybody who needs to hear that message is on your network, right? It's like sending a group email from work. Everybody who needs to know your communication, your colleagues that you're working with, everybody, like if I were to do it to you guys, I would need to make sure that all of you guys are on that group email to make sure that everybody hears the message. That means I don't just need a point source and a relay source, but I also need a network that touches all of you guys, right? So I need an email system that basically will bring all of you guys under the fold, in the fold, so to speak. These are the major pieces of your conductance system. So let's take a look at some of these and kind of dig in. So what we do is basically start off kind of a little slowly, and we kind of gently sort of wade ourselves into the pool. So we're getting kind of a little bit deeper with each slide until we're going to unfurl it into the full glory of the cardiac cycle. Okay. So first of all, these conducting cells that we mentioned before, number 3. These guys permeate the myocardium, but another thing they also do is they basically connect the SA nodes to the AV nodes. So this basically means that these two nodes communicate with each other through these conducting cells. And so they're able then to distribute communications through each other, and then they disperse that to the rest of the myocardium. So they're in between the two nodes, and then they also branch out into the myocardium. Now, in the atrium itself, remember you have two different types of cells in there, right? So you have the SA nodes who are conductive, and then you have all the rest of the cells in the atrium which are contracting. And so oftentimes what you'll have is what's called these internodal pathways. These are basically pathways of communication. That's between the nodes. So this is how the two nodes are talking to each other and coordinating their activities together. And that's what's happening in the atrium itself. In the ventricles, the way you form the ventricles is what's called the AV bundle, the atrioventricular bundle, which is essentially a branch of these conductive cells that sort of wraps itself around the ventricles. And this bundle will actually split into right and left bundle branches as they wrap around the right side of the heart, and then the left side will wrap around the left side of the heart. So this makes sure that both sides of the heart, right and left, are completely wrapped by this conductive kind of circuitry, if you will, that is formed from this AV bundle. So these are some of the major pieces of the conduction system. So you have your SA node, you have your AV node, you have your bundle and your bundle branches, and then Purkinje fibers as well. And so this is kind of what it's like. So these are your internodal pathways. So this is your SA node up here. This is your AV node. And so you'll see there's major communications between the SA node and the AV node, and you're basically kind of going all the way around. Now, there's a reason for that, because the signal for contraction comes from the SA node. Well, here's the problem. There's two things that the SA node has to do. And we're going to put this on the other slide, because this will explain a little bit about how the atria is wired. There we go. So from the SA node, it has to send a communication to the AV node, who sends it to the ventricles. So it's like a relay. So the problem is when the SA node says contract, it's got to do two things. It's got to send that signal to the AV node, but it also has to send that signal to the atrial myocardium as well, right? Because it's having to communicate this to everybody. And so it has two different pathways of communication. So in one pathway, it's got what's called intraatrial, which is basically communicating with other right atrial cells. So it's telling the right atrium to contract, but then it also has interatrial, which means it's communicating to the left atrium cells. So this message is going not just to the AV node, it does go to the AV node, that's one destination, but it's also going to the right atrial cells, and it's also going to the left atrial cells. So that message, once the SA node has it, is being sent in 3 different places. And that's important to understand, because what that means then is that both the right and left atrium are basically receiving that contraction signal at about the same time, because it's a very short distance and they're largely kind of together. So right and left atrium will hear the contraction signal, and so they contract at the same time. One of the reasons why both right and left are in unison is because they're getting the message pretty much at the same time. That's how quickly transmission is occurring between these connections. Now here's the problem. Moving to the AV node is going fast. So this is a fast transmission. However, from the AV node is slower. So the processing speed of the AV node is not nearly as fast as the SA node. That creates a little bit of a delay in the message because the AV node is a little slow on the response, right? It gets the signal from the SA node to contract, and the AV node is sort of like, you know, hanging out, just like, well, looks like there's something in my inbox. Should probably open it. It's from the SA node. Usually it's pretty important if the SA node sends it. I think I should probably open it, because if I don't read it, then something bad may happen. And a lot of you guys are like, just open it already, right? You're almost going to strangle me at this point. But it's like, okay, I guess I'll open it, right? Well, guess what? By the time the AV node gets around to opening that message, the atrial cells have already gotten it. So they're on their way to contraction. So a big reason for the delay between atria and ventricles is because of the slower processing speed of the AV node. That's how you decouple the 2 sets of chambers. That's the reason why atria and ventricles, all 4 chambers, don't go at the same time, is because the ventricles don't go until they hear from the AV node, and the AV node is delayed in its response. That's what creates that sort of discontinuity. Now, notice I said that everything is coming from the SA node. That is the SA node that gets the signals. As a matter of fact, in the previous chapter, what we also understand is that the SA node is basically spontaneous in its depolarization. So it's not that the SA node got the signal to contract. It's that the SA node generated the signal to contract. Notice the difference? Nobody told it to contract because it's automatic, right? That means that the depolarization is spontaneous. It's spontaneous depolarization. So ultimately what happens is, just like any other muscle cell, you're at resting potential, right, for these conductive cells. Eventually, you gradually depolarize, and then you hit threshold, and the cells are doing that on their own. So there's nobody doing that, right? Before, when you had that gradual, that graded potential, it was sub-threshold in muscular cells, it's because there was some other signal that was kind of slowly allowing sodium to leak in, right? There was something else that happened that allowed sodium to leak in, and that is the decision maker. That's something else that happened. Here it's happening spontaneously. It's constantly just sort of slowly seeping in. It's automatic, and then eventually it's going to hit threshold, and then it's going to open up the floodgates, and then you're going to get spontaneous depolarization, which then starts rapidly running through these conductive avenues, right, through these internodal pathways to the AV node and then to the other atrial muscle cells. Because of the automaticity of the SA node, it's called the pacemaker of the heart. Okay, so this is your natural pacemaker. And this is the one that sets your pace. So it's automatic. Now, does that mean there's no neural regulation on top of this? No, of course not. There's still neural regulation. It just isn't telling the heart to beat. All the neural regulation that's associated with this, right? Because we have vagus nerve, which was in charge of the rest and digest, right? The parasympathetic. And on the flip side, we had the sympathetic, which is the cardiac nerve, which stimulates an increase in heart rate. What those two nerves are doing is not telling the heart to beat. It's just either increasing the rate or decreasing the rate. The actual beating of the heart is determined spontaneously by the SA node, because that's your internal pacemaker. Okay. Once your heart turns on, it's on. Nobody's telling it to turn on or off. Now that you actually have your signal to the AV node and it's starting to process it, what it's going to do is then it's going to send that message into what's called the AV bundle. It's a little short strip of conductive tissue that it starts off in the atrium, enters the ventricular septum. That's one of the important things about the septum. It's not just the musculature septum, but it's also where you house your bundle situation. And then what's going to happen is the bundle is going to split into two halves, right and left bundle branches. So the right bundle branch will wrap around the right side and the left will wrap around the left side. And then it'll just kind of branch off into these smaller twigs and fibers of neurons called Purkinje fibers. So these are basically small conductive cells that make sure that all cells get the message. And you notice this is your moderator band right here in the right side. So the reason why it's part of the conduction system is because it has a little branch that goes through the moderator band, and then there's like these little Purkinje fiber twigs that basically make sure that you short circuit, you get to that lateral wall of the right ventricle so that you make sure that the right ventricle is contracting at the same time as the left. Why? Because the left gets everything, right? It's a dominant world. It's a left dominated world. So that means even your bundle branches are going to be more favored toward the left. So in order to make sure that your left and right are in synchrony, you kind of have a short circuit to help make sure you get that signal to the lateral wall of the right. Otherwise, if it has to go all the way around, it may not get there as at the same time. So what that would mean is you would have two ventricle chambers that would be slightly off pace with each other. So they'd be kind of contracting at different times. That, by the way, would be chaos in your heart. That would not be a good feeling in that particular case. So that's your conduction system. So when you take a look at this, then here's kind of the way the electrophysiology looks. So here's your threshold level at minus 40 millivolts. And so you can see then that spontaneously your pre-potential, right? This is spontaneous depolarization. I can't stress those two terms, those two words enough. Okay, a spontaneous depolarization. Nobody's doing this. It just is a natural kind of an ebb and flow of this. So naturally on its own, spontaneously, your your potential, your pre-potential, your greater potential will slowly increase until its threshold, and then you get that rapid depolarization, the same way we saw with the electrophysiology associated with skeletal muscle, with a couple of differences, right? And then on your recovery, you kind of go subthreshold, you hyperpolarize a little bit, and you kind of go right back to it again. Okay. So that's basically what's happening. And of course, it's the SA node that's essentially sending this message. Okay. And that's kind of an automatic thing. Now, let's take a look at heart rate. So your heart rate, obviously it can vary quite a bit, right? I mean, at rest, your heart rate can be quite low. And during exercise, your heart rate can be quite high. So that's an important thing, right? That seems like a very benign piece of information, right? Like a very easy kind of a low bar. Wow, I hope that's an exam question, because that's going to be an easy one to get, kind of a question. But it couldn't be more important. Because when you take a look at your heart rate, a lot of times students will be like, well, wait a minute, how come there's like so much variation in your heart rate, right? How come it can fluctuate to like really, really low when you're really chill and relaxed, to really, really high when you're really stressed or working out or something like that? And the answer is it's designed that way. Why? Basically, it's designed to essentially change with your demands. Think about it for a second. Everything about your cardiovascular system is basically rigged like an economic system. You have a product, and you have demand and supply. Right? What's the product of your cardiovascular system? What? Okay, that's fine, but what's the actual product? Why do you need blood flow? Okay, but what do you need in order to stay alive? Oxygen, right? And of course, nutrients go along with that, right? That's your product. Oxygen in particular, right? So why do you need oxygen? To stay alive, right? Because you need energy, because the only reason you breathe oxygen is so that you can feed that to your cells. And what are your cells using that for? It's using it to go through cellular respiration in your mitochondria, in your cells, which makes ATP, which is your rechargeable battery that allows you to be able to have the energy to do any kind of a biological or cellular process in your body. And there's a lot of them. So essentially, ATP is like your cell phone battery. It's rechargeable, but you need it if you want to be able to have all of the rich capacity and abilities that your cell phone gives you, complete with the entire world, the app stores and all that sort of stuff, right? You can pretty much do just about anything with your phone. Actually, most of us probably do more with our phone than actually call. It's like, oh yeah, your phone does all this, and you can also call with it too, right? It's kind of like it's outgrown its original use. It's more of like a personal communication device than an actual phone, right? But that's your product, oxygen. But guess what? Do you always need the same amount of oxygen all the time? Because remember, the termination point for oxygen, the whole point of it is to generate ATP, energy. Right now, are you using a lot of ATP? No. So do you need a lot of oxygen? No. How do you know you don't need a lot of oxygen? Just check your breathing rate. Are you guys huffing and puffing? No. Why do you huff and puff, by the way? To get more oxygen. Why? Because who needs it? Your cells. That's the demand. What is the supply? Your respiratory system. So the exact same ideas that drive economics in terms of supply and demand. And if you ever study supply and demand, it's actually fascinating to study it, right? Because it's always oscillating. It's always kind of fluctuating, right? Demand will rise really high, and then supply will have to match it, right? And then demand will drop, and then supply will drop, right? So if you get into economics and get into like retail, you'll notice that there's an entire branch of retail called inventory control. Because the idea is you want to have enough inventory on your shelves in order to meet demand, but not so much that you have a glut of excess inventory, because then you're going to have to mark it all down and toss it on the clearance rack to get rid of it, right? So you're always trying to match your supply to your demand, but guess what? Your demand is always changing. Same thing's also true for your body. As your demand changes, your supply has to change. Well, that's all well and good, but what does that have to do with the cardiovascular system? Last time I checked, we were talking about breathing. That's the respiratory system. It's a few chapters away. So what does that have to do with anything? Well, think about it for a second. If you've got a supply of product, how is it that you actually get it into the hands of the consumers? Do the product fairies just magically materialize it in front of them? No. You have to make it, right? Most of the time, you have to distribute it. And there's an entire industry in economics that a lot of people are ignorant of, the entire distribution system. We see it as shipping, right? But shipping is only a small part of distribution. Most of distribution is associated with actually moving your product to the points of demand. And how do you do that? You have to create an entire system, right? I mean, think about it. How many of you guys have seen a big old 18-wheeler Walmart truck rolling into a Walmart? That's distribution. They didn't make that here in Denver. That was probably made in their manufacturing outlets in Arkansas. So Walmart has to have a distribution system to get that product on the shelves here in Denver so that it can meet the demand of Denverites. So what does that mean? The cardiovascular system is your distribution system. Think about what you need for distribution. You have a product, got it, okay, got that, done with that one. But now what do you need in order to move it from Arkansas to Denver? You need something to load it onto, right? In this case, the Walmart truck, right? So you need something that will physically carry it to its new location. Guess what that is in the cardiovascular system? We just studied it. That's veins and arteries. We're going to get to that one. The actual physical trucks that are carrying the product are what? The red blood cells, and specifically the hemoglobin in the red blood cells, because they're the ones who actually are loaded up with the oxygen. So your red blood cells and your hemoglobin are essentially the Walmart trucks that are carrying the product. So now let's get down to the roads, right? Because that's kind of what you mentioned, right? So what about the roads? What are the roads in the cardiovascular system that the trucks have to drive on that get you from Arkansas to Denver? Those are the blood vessels, right? So the blood vessels are the thoroughfares or the roads that the trucks, the blood cells, can drive on in order to deliver your product to the point of demand. But guess what? There's more than that, isn't there? Because you can regulate that journey, can't you? Now, in our particular system, our trucks aren't self-motivated, right? I mean, they're not like a Walmart truck. They don't basically drive themselves. Some of them do. There are actually some self-driving trucks. It's actually creepy and cool at the same time. But that's not what red blood cells do they? So you actually have to create motive force in order to get these things to move from point A to point B. And that's where the heart comes into play. So the heart is like the primary central distribution center that pushes literally all of the product onto the red blood cells and out through the blood vessels or the roads of the country to distribute them to different places, different states in the union. Everything about the cardiovascular system is basically a gigantic distribution system. And just like Walmart, it is as permeated into society or in terms of physiology as Walmart is. I mean, Walmart is everywhere, right? I mean, there isn't a place that's untouched by Walmart on this planet. I mean, they probably have Walmarts in New Guinea where they're still eating people. They'll probably sell shrunken heads there. Great value shrunken heads as opposed to brand shrunken heads. Try them. You'll like them. They taste as good. Whatever. But you know that's true. It's probably right next to the McDonald's serving the People Mac instead of Big Mac. Anyway, okay, I'll stop. But that's basically what you're talking about here. So you're talking about moving product, and this is the major way you move it. It's a major hub of distribution in order to disperse product throughout the rest of your body, and that's your focus. So why is it important that you have such variability in your heart rates? Because your distribution always has to match your demand, which means when you're at rest and you're chill, you don't have a lot of demand, so your heart rate is low. When you're running and you're working out, your demand is much higher. So you have to increase that distribution and everything that goes with it. Okay. So when you take a look at that then, and you take a look at heart rate specifically, the SA node basically, when you're looking at it, is on a per minute basis, it's usually firing 80 to 100 action potentials per minute. This is what the SA node is doing by itself. This is its processing speed. Now, this is its like it's more of this automatic. So this is that automatic rate. It kind of gives us a little bit of a starting point to kind of tether ourselves to. Obviously, the parasympathetic stuff, like the vagus nerve, for instance, will slow that rate down, but it will not. It doesn't tell the SA node to contract, right? It doesn't tell it to do that. It just kind of slows it down a little bit. The AV node, in comparison, is looking like about half that, right? So it's like about half as fast as the SA node. So it's spitting out about 40 to 60 per minute, which basically means that this is a slower processor. And the reason why there is a delay in conduction to the ventricles. So when we said that it's atria ventricles, atria ventricles, atria ventricles, the reason for that is because of the disparity in processing time between the SA node and the AV node. Okay, so let's take a little bit of a deeper look and we'll kind of wrap this all together and kind of see a little bit of cartoon sketch here. So you have your sinoatrial node, your SA node, which is your pacemaker, right? This is the one that starts it all off. And so this is going to be the posterior wall or the back wall of your right atrium. So this is your pacemaker cells. These are those conductive automatic cells. And so they will be conducted, or they'll be connected rather to the AV nodes with these internodal pathways so that they have communication to the AV node as well. And these are the ones that begin it all. They start it all off. They basically initiate action potential in step one. So it's kind of what it looks like. So here you can see this little purple right there. The SA node is now activated. So it is now sending out an active action potential, a pulse, essentially throughout the rest of the heart. And here you can see the AV node here, and you can see the bundle system going through the septum. Now notice one thing, this is happening at time 0, right? This is where we start the clock. This is at time 0 where you have the activation of the SA node. And then what's going to happen is from here, it's going to go to the AV node, right? So this is going to be still in the right atrium, but it's on the bottom floor. So it's inferior to the SA node. Like that, nice use of the lingo there, right? So it's going to be on the inferior side of it. It's going to release through these internodal pathways, the SA node's communication. That's going to be step 2. But because of its processing speed, remember, we're talking about 40 to 60 action potentials per minute, so it's slower. So that slower is going to basically delay the relay of that to the ventricles. So while it's slow in its relay, the atria are already contracting at this point. So when the atria are contracting, the signal now is in the lap of the AV node. And it's getting ready to pass that on to the next point, which is the AV bundle. So this is what it looks like. So right now the AV node has the signal, but you can see this wave of purple. That's the depolarization of the muscle where the atria are now starting to contract. So you can see that, first of all, you've got this wave where you have communicating to the right atrium cells, but now you're also communicating to the left atrium cells as well. So there's a wave that basically covers both atria, and this is going to be at about 50 milliseconds down the road. Now, next step. Contraction will then basically roll through the atria. So here you can see the AV bundle actually kind of receives it. So actually they cut this up into two parts, right? So we kind of covered the end part. So they covered this in two parts. Before the AV bundle gets it, the wave of contraction is already beginning. So the message is starting to move through this internodal pathway, but the AV bundle doesn't have it yet. Because if you think about it, it's like a tsunami. If you've ever seen a tsunami, that wave just comes in and it keeps coming and keeps coming and keeps coming, right? But generally speaking, it's all coming in at the same pace. So if you're standing here, you're going to get hit by the wave at the same time as if you're standing here. What that means is that this signal is like a tsunami wave. It's basically hitting all of those atrial cells at the same time and at the same rate that it's progressing toward the AV node. So the AV node doesn't get it yet because the wave hasn't hit it yet, but the atrial cells are contracting and they're beginning to contract. Okay. So you can't, so I guess I say that because to say this, that you can't really blame it all on the AV node. It is slower, yes, but before it even gets it in the inbox, the atrial cells are already contracting because they've already got the message. Okay. And then as that flows through, then the message reaches the AV node, but by that time, your atrial cells are already on their way. So you can kind of see the walls collapsing. So that's contraction. So depolarization has already engulfed both atrial chambers, right and left. And now they're starting to contract together in unison. Okay. And while they're contracting, the AV node now is slow in the process. And so it's still in the inbox of the AV node. And then the AV node is slowly starting to send that forward. So you can see here, it's moving into the AV bundle. Or sometimes this is referred to, you'll see this term as well, the bundle of His. That's oftentimes what it's called. And then this is happening at about the 150 millisecond point. So now at this point, atria have already depolarized. They're already contracting. They're in the middle of contracting. The action potential is now just starting to move its way through the bundle system. Right, starting off with the AV bundle, and then it's going to branch and separate into the two bundle branches. So in the septum, it's going to be moving into the AV bundle, and it's going to split into right and left bundle branches as it wraps around the lateral walls, and it's going to spread into these Purkinje fibers, these like little fingers of conductive cells that sort of permeate all the nooks and crannies of the lateral wall. And they're also going to be moving into the moderator band. And the moderator band on the right side is what basically controls the papillary muscles. Because one of the things you need to understand is, like, where we talked about the papillary muscles hanging on to the chordae tendineae to keep the AV valves from inverting. But what actually happens is when the ventricles contract, the way that they hang on to the tips of the chordae tendineae is they contract themselves. And it's a little bit offset. So as the ventricles are contracting, the papillary muscles have a little, just a very slight delay. And so what happens is, as that pressure builds and it starts to want to go up through the AV valve, what's going to happen is those papillary muscles contract, and that basically grabs on to the tips of the chordae tendineae, keeping the valve from inverting. Right? That's basically how that works. And then the Purkinje fibers are doing the same thing on the left side. Okay. So the Purkinje fibers in the moderator band are kind of synonymous in function, but because you've got all the Purkinje fibers dedicated to the left side, the moderator band is created to help with the right side. You can see your wave is now going down your septum. It's gone into the two branches and into the moderator band and through the lateral wall. So this is happening at about 175 milliseconds at this particular point. As it wraps around the left side, it's then going to start to move through into the Purkinje fibers, which is all about distribution through the ventricles so that every nook and cranny gets the message. So by the time this happens, atrial contraction is done. And now the atria are ready to relax, but ventricular contraction is just starting. So you don't have, it's not like it goes atria, ventricle, atria, ventricle. They're literally right on top of each other. It's like atrial ventricle, atrial ventricle, boom, 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 boom. So there's not a lot of lag time in between when the atria stops and the ventricle starts. So the Purkinje catheter, here you can see now the wave is wrapping around the lateral wall. It's already in the ventricular septum. So you're getting contraction signals from the center, and you're also now starting to get contraction signals from the lateral side as well. And that wave is starting to move in this direction. It's bouncing back in this direction, and you're getting a contraction from the bottom up. So the ventricles actually contract kind of like from the bottom up, almost like you're squeezing from the bottom, and you're kind of rolling it upward. So you're kind of like squeezing it from the bottom. Okay. So that way on the ventricular blood, forcing it forward. This is happening at this particular point once the ventricles are bathed in depolarization and they're starting to contract. This is happening around roughly or so the 225 millisecond. So the reason why we do that one is because, think about this for a second, the amount of time that it takes to go from the firing of the SA node to the complete, now you're not completely contracted, right? But the complete delivery of the depolarization event, because remember, we're not looking at contraction in these slides, we're looking at depolarization, we're looking at the conduction of it, right? The signal. It takes less than 300 milliseconds from start to finish. So oftentimes that will kind of cause some pause in students, but they'll be like, well, wait a minute, if there's like this big delay, I mean, isn't that a bad thing? I mean, is that going to be bad for the heart? Well, no, not really, because the delay that we're talking about is less than 300 milliseconds. So it's still, all of it together, including with the delays, is still a very fast process. And if you're wondering, just to give you a sense of scale, a millisecond is 1,000 seconds. Okay, so that's what we're talking about, just in case. Excuse me, it's 1,000th of a second. It's not 1,000 seconds. I went the other way around, right? So it's 1,000th of a second. Okay. 0.001 seconds. So what about pacemaker abnormalities, right? So these are basically oftentimes what we refer to as arrhythmias, which are essentially abnormalities of various sorts in the conduction system. And an arrhythmia technically can be a pacing problem. That means it's an SA node issue, but it can also be a conduction problem. That was a different thing. It's not quite the same thing as pacemaking. So this is a particular type of arrhythmia that was associated with pacing. And so remember, the operative word here is abnormal. We already know that pacing of the heart is designed to be plastic. It's designed to have some plasticity. So it's not abnormal to have a low heart rate. It's not abnormal to have a high heart rate, right? So we have to define what we mean by abnormal. Now, generally speaking, if you have a low heart rate, it's called bradycardia. Slow heart rate is called bradycardia, but it has to be abnormally slow, not normally slow, right? The other side of that is tachycardia. So this is an abnormally fast heart rate. But notice again, the operative word is abnormal. So what do we mean by that? So if I have a low heart rate that's really low and I'm taking a nap, is that abnormal? No. If I have a low heart rate and I'm trying to run, is that abnormal? Yeah, right? If I have a high heart rate and I'm working out, is that abnormal? No. If I have a high heart rate and I'm just sitting down, is that abnormal? Yeah, right? Notice its abnormality is associated with it's like a dysregulation between what your body is doing and what you're doing, right? We already said that your body, the reason why you have plasticity in your heart rate is because your cardiovascular system has to be able to respond to changes in your body's demands. Now, when you're talking about abnormal, it's when your heart rate changes in an abnormal way. Demand has just gone up, but your supply is low. That's an abnormal response, right? Or vice versa. That's when we start talking about things like bradycardia or tachycardia. One of the most common things that patients make, the mistakes that patients make, is they will, go to Walmart or whatever, and they'll take their blood pressure and it'll be high, and they'll freak out, and they'll start calling it all sorts of doctors, right? Or they'll they have these like smart watches that are measuring all sorts of stuff, or these apps where you can measure your blood pressure and your heart rate and things like that, and it'll be like really high. And then they'll freak out and they'll go to the cardiologist's office. And typically a cardiologist will basically look at you and like, why are you here? Right? Because usually patients don't make that association that it's not just your heart rate or your blood pressure. It's about your heart rate and your blood pressure and what you're doing. Of course, you're going to have a high blood pressure if you're at Walmart. You're at Walmart, right? Nothing good happens at Walmart. It's like a black hole for humanity. Everything stressful happens at Walmart. You know this is true because there's probably more YouTube videos on Walmart than any other industry company out there, right? Bad things happen at Walmart. Of course you're stressed out because you just came to get one thing and you know you're going to be there most of the day trying to get the hell out of there with your one thing. So that's natural. Or like you take your blood pressure at the doctor's office, like, oh my gosh, my blood pressure's high, my heart rate's high. Of course it is. Because you're afraid of doctors, right? So don't talk to me about your heart rate being high or low until you tell me what you're doing. If your heart rate is like 120 and you're taking a nap, yeah, you probably need to go and consult a doctor on that one. That's not a good combination, right? Your heart rate should make sense to what you're doing. Okay? That's what we're talking about now. The major abnormal one is what's called ectopic pacemaking, which is also a bad one. And so ectopic pacemaking is a situation where you have non-SA cells initiating an action potential. Now, the reason why this is a problem is because abnormal cells, these are the non-SA cells, are initiating an action potential. And because they're doing it, the SA is still doing its job, so it's going to increase the rate of action potentials. Oftentimes, it will bypass the conduction system, so it's not following the normal routes of communication the way you've set up, and it can also screw up your ventricular contractions. Think about it this way. Everybody's right for the SA node because they're all like waiting. It's like, okay, as soon as the SA node says fire, then that's going to ripple through the entire system, and then we're waiting kind of for when we get it, and then we go, right? And so everybody's waiting. Now, imagine if you had multiple patches of cells that were competing voices with the SA node. So instead of hearing a singular contract, 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 you started hearing this sort of muffled background, like contract, 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 no, contract, 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 contract. Who are you following? Right? So it creates a lot of contrast. Some people are going to follow the first one. Some people are going to follow the second one. Some are going to follow the third. You're going to get all these sort of random, sort of disharmonized signals coming through. The ventricles aren't going to know what to do because they get these sort of random choppy messages of contraction because they're hearing all these sort of voices in there and they're not really able to focus on the voice that they need to listen to. And that creates a lot of what will create ultimately is kind of a lot of misfiring and a lot of miscontraction in the ventricles. And if they're hearing a lot of this, the ventricles will themselves kind of try to contract every single one of them. And it'll kind of start to create like ventricular quiver. It'll be like shivering. It's like, it'll be like contract, contract, contract. But it won't be giving a full contraction because it won't be allowed enough time to reset itself. That's what the SA node does. I say contract. Reset. Now contract. Reset. Now contract. Right? But if you have all these ectopic voices, you're never going to get to the place where you reset because the SA node holds off, knowing that you need time to reset after you contract. And so that creates a lot of ventricular disruption. And sometimes the ventricle just takes things in their own hands and they're like, you know, screw this. I'm just going to boom. I'm going to fire one off. because I don't know what you guys are doing. So I'm just going to pick something and go with it. Generally speaking, if you've got ectopic pacing, usually in cardiology, they'll have to go in there and they have to do what's called an ablation, which essentially means they kind of go in there with it's a cath lab visit usually. And what they'll do is they'll go in there and they'll actually just burn off, literally ablate those ectopic cells. So they'll kill them, because the idea is like, listen, you're not doing your job because you're not supposed to be the one in charge here. It's the SA node. So we're going to have to kill you, because it's better that you die than that you create this kind of chaos throughout the heart. Okay. Pretty severe, but that's basically the way your heart handles it. So let's take a little bit of a closer look at our EKGs. So we already said this is basically recording the electrical events. Notice not the contraction. The contraction follows, but in reality, this is the change in voltage. That's what you're looking at. So you're looking purely at the conduction system. And so you set up an EKG by establishing electrodes across the body, right? There's different ways to set this up. There's like the traditional 12 lead and things of that nature. But basically what you're doing with the electrodes is you're setting them up in opposition to each other, right? So you set them up so that there's like a reference electrode that basically will see different types of voltage changes as the heart goes through its conduction system. That's the reason why you have all these different leads all over you, is so that you can kind of get all this information from these different electrodes, and they kind of essentially see slightly different things from different angles. Okay, kind of like having somebody tell the story of a particular event, but having everybody tell their own story, and everybody has a slightly different version of the story. That's because they all saw it from different angles. But if you take them all together, then that's kind of the whole story. And that's kind of what you do with these electrodes that you put on the individuals. They can also be used actually quite expertly to be able to diagnose damage. A lot of people don't realize that. But when you take a look at a good EKG, a good well-trained cardiologist can actually do a lot of diagnostic work from an EKG. And in cardiology, I feel like EKG is probably the most underused technique that we actually have. Not in cardiology. Cardiologists use it all the time, but in general medicine, it's super easy to run. It's not difficult at all. It's super non-invasive. Most people have a problem with it, and you can get a lot of information from it. The problem is most primary care physicians don't have a clue what to do with it. I know that because they used to send us their EKGs all the time for our doctors to read and then give them results back. But you can get a lot of information off of this. Now, when you look at an EKG, there's certain waves that are associated with different events that are happening during the conduction cycle. So when you take a look at your basic EKG, you're going to have different waves to it. And I'll write these guys here in just a little bit. So basically you have your what's called your P wave. And then it kind of goes through this sort of big complex sort of QRS wave. And then there will be what's called the T wave until you get back down to normal. So this would be P, this is QRS, and this is your T. What's happening in each one of these? In the P wave, this is being read, your atria are depolarizing at this point. So that's what it's reading. It's reading the voltage changes associated with atrial depolarization. In the QRS, your ventricles are depolarizing, but also your atria are repolarizing. So you're reading both. That's the reason why it's a more complicated curve, because there's two different things that are going on that are overlapping. And then the T wave is basically when your ventricles are repolarizing and getting themselves reset again, so you can do it all over again. And those are the basic components of an EKG wave. So when you take a look at it, you have these basic waves. So here's your P, your QRS, and your T wave. Now, things you can tell from this, certainly you can tell things like heart rate and stuff like that. But you can also tell if there's different problems associated with the conduction system of the heart. So for instance, with the PR interval, and I want to fast forward to this. So this is your PR interval right here. This is the distance from the beginning of your P wave to the beginning of the QRS. So this is largely looking at dysfunctions, if you will, of atrial depolarization, right? So if your atria are having a hard time depolarizing, this PR interval will lengthen. And a long PR suggests atrial depolarization defects, okay? Now, the other one that we have is the QT interval. This is a big one. This is looking mostly at ventricular depolarization. That's primarily what it's looking at. But when we take a look at the QT interval, it's the beginning of the QRS curve to the end of the T. So this actually is both depolarization plus repolarization of the ventricles. So this is an important one, right? Because if your ventricle, and by the way, let's be honest, what we're really looking at is the left ventricle here, right? So if your QT is long, then that suggests there's some sort of an abnormality, that your left ventricle is having a problem depolarizing sufficiently. That means there's an issue maybe with the bundle branch, the left bundle branch in particular, or it's having a hard time repolarizing. That basically means it's either one of two things. It's either having a hard time depolarizing, which means it's having a hard time contracting, right? And it's repolarizing just fine. Or it's the opposite. It's contracting just fine, but it's not really relaxing very well. You can actually separate these two by doing what's called an ST interval. So this one is just going to be the repolarization of the ventricles. So you can separate whether or not your ventricles are struggling with depolarization or repolarization by either looking at, first of all, the QT. Long QT is a bad thing, by the way. That's a sign of a heart that's in failure, okay? And you can compare it to the ST, because that way you can distinguish between a repolarization problem versus a depolarization problem, okay? So when you compare QT and ST together, that's how you separate those two out. And so those are the really two big lengths. And oftentimes a good cardiologist can actually diagnose you based on that. So here's kind of what it looks like when you get the lead set up. And so you can see they're just basically measuring electrical changes throughout. And so this is your basic EKG. And what I want to do is I want to finish up to here. That's where the exam is going to stop, right there at the end of the EKG section. Okay. These are some patterns of EKG that are somewhat abnormal. The only thing you need to do is just basically be able to read an EKG and tell me if it's abnormal or not, right? Because this is normal. This is what a normal rhythm looks like, and these guys are a little bit abnormal. So the first one is what's called premature atrial contraction. So what does that mean? That means normally you have a normal P, a QRS, and here's your T. Here's a P, QRS, and here's a T. But now instead of a normal rhythm, boom, you get a quick P in there. So this is basically an abnormal P. So it came a little too quick, okay? And then you get a normal rhythm after that. So it's, once your P starts, you get a normal QRS and a T. For a paroxysmal atrial tachycardia, this is basically a atrial spiking, right? An atrial runaway is what it is. So here's your P, QRS, and your T. Here's your P, your QRS. But notice what happens is you have a rapid fire. Your P and your T overlap. And so now you're just getting this boom, 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 this rapid fire, because you have this abnormally quick P that comes in before you have a chance to relax, right? Before you get a chance to repolarize. And you see that it's kind of like a rapid succession of these ventricular spikes in there. Atrial fibrillation is basically atrial quiver. This is what happens when your atria are just kind of like rippling like that. They're not really getting a strong unified contraction, just sort of rippling. And so here you can see, here's your P, here's your QRS, and here's the T. But then notice you kind of get this sort of atrial ripple. And the ventricles don't know what to do with that. They're like, it's like a crowded room. It's like, roll, 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 roll. And so the ventricles be like, you know what, I'm going to go on that one, go, boom. And then they're roll, 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 roll. I'm going to go on that one. Okay, go, boom, right? So they kind of have to make it up as they go. But what you have is a sort of atrial quiver, not that nice unified. Premature ventricular contractions, PVCs, is essentially similar to the atrials, only now it's for the ventricles themselves. So here you have your P, QRS, and your T. But then notice, instead of a P, boom, you get a ventricle in there. So that's a ventricular contraction. That's a ventricular depolarization. And notice you also get a deep trough right afterwards usually. And so PVCs will occur every now and then, like isolated, boom, like just like rogue waves, if you will, from the ventricle that just happened. But otherwise, if you maintain normal rhythm, you're good. Usually if these recur commonly, then usually a cardiologist wants to keep track of them, right? Because that's considered a dysregulation of your rhythm. Ventricular tachycardia, this is basically where the ventricles are firing instead of the atria. So here you have your normal P, QRS, here's your T, but now all of a sudden it's like boom, 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 boom. So this is basically over and over again. So this is basically where you have an abnormally fast ventricular contraction without being aligned with what's happening in the atria. Okay. And then of course, ventricular fibrillation is essentially ventricular quiver. And this, by the way, is a very bad sign. This essentially means your ventricles are quivering and they're not able to do their job, which means you're not getting any blood out of that left ventricle. It's not getting to your brain. It's not getting to your lungs. It's not getting to your body. It's not getting anywhere. This is basically the sign of somebody who's dying when you see ventricular quiver. This is when you pull out the paddles and zap them to get your heart back into rhythm. Okay. This is usually due to gross damage of the heart in the conduction system. So you can't maintain rhythm. That's basically what happens. Okay, so that's the cutoff, and then we'll start next time with the contractile cells after the exam. As you guys flee screaming. And that part is recorded.